Let us now dig a deeper into the process of fertilization. Now, when the sperms are being introduced into the female genital tract, that is, we call the process as insemination. After the insemination is done, the sperms travel through the vaginal tract of the female at a speed of about 1.5 to 3 millimeters per minute. And out of those many sperms which were released, only few are capable enough to reach or travel right up to the fallopian tube, and they will be fertilizing the ovum. Also, when the part of the ovum which receives the sperm is known as the animal pole. From this part itself, the polar bodies are extruded. Also, the other pole will known be as vegetal pole. So the ovum will be having the vegetal pole and the animal pole. And the animal pole is the region where it receives the sperm. Now, after the sperm enters or it reaches to the ova, following events occur in a series, and these all events we are calling it as physico-chemical events of fertilization because it will involve many physical and chemical changes. We'll be discussing one by one. The first is the acrosomal reaction. Now, we have studied the structure of the sperm where the head has the acrosome structure carrying certain enzymes. And now these enzymes play a very important role in bringing about the fertilization. So the acrosomal reaction involves the acrosome area of the sperm consists of certain sperm lysins. These are enzymes, and the various enzymes over here which are present are, first is hyaluronidase. Hyaluronidase, it dissolves the hyaluronic acid of the follicular cells which are present in the ovum. So this acid or this enzyme will help in the dissolution of the hyaluronic acid of the follicular cells. The second enzyme which is found is corona penetrating enzyme. The corona radiata layer which is present outside the ovum, outside the zona pellucida, needs to be removed. So this enzyme, that is corona penetrating enzyme, dissolves the corona radiata, the outer layer of the ovum. The third is the zona lysin or the acrosin. These three enzymes, this zona lysin dissolves the zona pellucida layer, which is again the inner side. So if you remember the diagram, the outer side, this is the corona radiata layer, which is present outside the ovum. So this corona radiata which is the outermost layer, and it is when the sperms reach over there, the corona penetrating enzyme will dissolve this layer. As a result, the sperm will be able to move further. Now this layer is the zona pellucida layer. This zona pellucida is also that it is dissolved with the help of the enzyme acrosin or zona lysin, and the hyaluronidase dissolves the hyaluronic acid of the follicular cells. So the first reaction is the acrosomal reaction. As soon as this is the plasma membrane of the egg, plasma membrane of ovum. Now, as soon as the sperm reaches over here, there are chemicals, that is fertilizin and anti-fertilizin. The fertilizins are present on the ovum, and the anti-fertilizin is present or it is produced by the sperm. The fertilizin is a glycoprotein and the anti-fertilizin is a acidic protein, and the combination of these two that was discovered, the process was discovered by F.R. Lilly, and this enabled the reaction between the fertilizant and the anti-fertilizant. That is, the fertilizant is found to be present in the ova, whereas anti-fertilizant is found to be present in the sperm. This is acidic protein, and this is a glycoprotein. Now, both of the chemicals react, and the process was discovered by F.R. Lilly. Now, both of them react, and as a result, the sperm is able now to attach its head to the cell surface of ovum for the process of fertilizing. So, fertilization is brought about with the help of fertilizing and anti-fertilization, where the reaction takes place, and as a result, the sperm becomes now capable, or the entry of the head of the sperm is possible. The third is the dissolution of the plasma membrane of the egg and the sperm. As soon as this reaction between fertilizing and anti-fertilizant take place, the plasma membrane which is present over here dissolves, and the sperm will now be capable, or the head of the sperm will enter into the ovum, leaving the tail behind. Now, over here are the cortical granules. These are the cortical granules which are present on the egg. This is the nucleus. If we draw the complete diagram, over here, this is the cortical granules on the sides, and this is the egg nucleus, or we say nucleus of the ovum, which is eccentric, already discussed, towards one side. Now, as a result of the entry of the sperm head inside the cytoplasm, a cortical reaction takes place, or we call it as Zona reaction. Now, what is meant by that? The cortical granules which are present in the cytoplasm of the ova, they fuse with the plasma membrane of the egg and release enzymes so that the zona pellucida is hardened. So as soon as the contents of the sperm enter inside the cortical granules, which are actually proteins that react with the zona pellucida layer and release certain enzymes that harden the zona pellucida, and it causes the depolarization. As a result, the entry of the other sperms is prohibited or is avoided. So that is what is we mean by polyspermy is prevented or polyembryony. Polyembryony means formation of many embryos. So this is prevented because at a time or during the fertilization, only one sperm is going to enter inside the ovum. So the reaction of the cortical granules with the zona pellucida results into the depolarization of the ovum, first thing, and the second thing, it causes the hardening of the zona pellucida layer. Due to this hardening of the zona pellucida layer, no other sperm which is approaching over here will be allowed to enter and break through this, and already one of the sperm has poured its contents inside the ovum. Now, after the reaction this has taken place, this triggers off the entry of the sperm head inside the ova, triggers off the second meiosis to complete. We have studied that the meiosis is rested at a stage, and at that point of time when the sperm enters inside, this triggers off the completion of the second meiosis, and as a result a larger cell called ooted is formed, and a smaller cell which is the second polar body now is being formed, which gradually degenerates, leaving only the ooted behind, which is the large cell, and this cell has most amount of or maximum amount of cytoplasm. Now, after the completion of the meiosis, that is the meiosis 2, the male nucleus will convert into pronucleus and it will fuse with the egg. This nucleus will fuse with the egg to form the zygote after the process of fertilization. So the male nucleus fuses with the female nucleus. The process is known as fertilization, and this will result into the formation of zygote, which is now a diploid structure. So the zygote formation takes place after the series of reactions which are taking place. Let us try and understand and go back again. Soon after the insemination, that is the introduction of the sperm inside the genital tract of the female, the sperms in millions travel through the genital tract, reaching to the oviduct, actually where the fertilization process will be taking place. And once the sperms reach over there, only few reach to the fallopian tube or the oviduct. The egg at the same time is being released by the process of ovulation, and it is traveling through the infundibulum and reaches to the ampulla isthmus region. Ampulla is the region where the fertilization will take place. So the acrosomal reaction, due to the action of these enzymes, that is hyaluronidase, corona penetrating enzyme, and zona lysine or acrosin, the various layers of the egg are broken or degenerated, and due to the reaction of fertilizin and the anti-fertilizin, the fertilizin is a glycoprotein which is being produced by the ova. Anti-fertilizin is a acidic protein which is being produced by the sperm. These were discovered by F.R. Lilly, and these two enable the sperm to enter or to attach itself to the cell membrane of the ova for fertilizing. Now, after the attachment of the sperm to the ova, the plasma membrane starts dissolving, and as a result, this head of the sperm enters inside the ova, leaving the tail region outside. This also triggers off the cortical reaction, in which the cortical granules will react with the zona pellucida, and it will also cause the depolarization to prevent the entry of other sperms, because due to this reaction, the zona pellucida layer, which was earlier, before the entry of the sperm, it was earlier ready to take the sperm, now hardens, because only one sperm can enter at a time, and therefore this, after entry of the one sperm head, that or the contents of the sperm, the cortical granules will react and will form a hardened layer. Now, as a result, the polyembryony or polyspermy is prevented. This also triggers off the process of meiosis II to be completed, and as a result, a larger ootid will be formed and the second polar body will be formed. The larger ootid will later on fuse with the sperm, and the second polar body will be released or degenerated. This causes this fusion of the male and the female nucleus is known as amphimixis, because the male and the female two are getting mixed up, and this results into the formation of the zygote, which is a diploid structure. Now, the later on, this fusion has several importance, because the process of fertilization ensures the maintenance or restoration of the 46 chromosomes. That is, the number of chromosomes is fixed, and also it maintains the variation. So fertilization is an important phenomena, as it also decides the sex of the baby. So now in the next part, we are going to discuss about how the sex of the baby is determined, and also the implantation and further embryonic development.